For billions of years, life on Earth operated using a very specific set of rules. Rules encoded into our DNA and evolved through natural processes. But what if we could begin to rewrite some of these rules, eventually allowing us to not just edit them, but truly engineer life from scratch? Or even create something that seems to be almost life, but that's not entirely life yet. And that's the ambitious goal behind what's known as synthetic biology. And so, hello info person, this is Anton, and in this video, we're going to explore some of the recent and somewhat groundbreaking discoveries, essentially highlighting how modern biologists have been pushing the boundaries when it comes to this field in order to help us understand the living systems and in order to uncover a lot of unexpected properties of life in the process, but specifically moving beyond genetic modification and tweaking existing organisms and venturing into the creation of novel biological entities capable of things never before seen in nature. And specifically in this video, we're going to be focusing on three main types of discoveries: the cyborg cells, the newly designed bacterial cells, and the advances in synthetic biology when it comes to much more complex cells, such as the ones inside of us. And so let's talk about these studies in a little bit more detail, and let's start our journey with the study right here. The fascinating concept explored in this study that the scientists in the study decided to refer to as cyborg bacteria. And so what exactly is this and how does this work? Well, typically, synthetic biology involves either actual cells or natural cells, which are complex but are difficult to replicate and does actually raise safety concerns, or artificial cells, which might not be able to replicate and generally have reduced biochemical complexity. You can actually learn about these synthetic cells created in some of the previous studies in some of the videos in the description. And so the challenge has always been the same, trying to combine the strength of both: versatility of natural cells with the control and defined functions of artificial materials. Mostly because this would be used in research, such as for example trying to create new drugs or trying to understand how certain cells react to certain conditions. And so here, for the first time ever, researchers were able to create a hybrid material. And because this is a hybrid, they decided to refer to them as cyborg bacteria. And the main idea here was to try to assemble a polymer network inside a bacterium like E. coli, and this would prevent it from dividing and from replicating. And the first assumption was that this would actually kill the bacteria, essentially making the whole thing useless. But unexpectedly, these strange cyborg cells preserved some of their essential functions, including cellular metabolism, motility, and protein synthesis. But they were no longer able to divide. So these were basically semi-living entities. They had most of the living functions, but not division. And the main process was pretty simple. Here, E. coli were infused with synthetic hydrogel, mostly made out of polyethylene glycol, along with the photoinitiator and the fluorescent dye, in order to track their progress. And this was achieved using a single freeze-thaw cycle and the washing away of various extracellular components in order to eventually create perfect cyborgs. And here researchers tried to achieve 5% hydrogel density in order to form a kind of a porous scaffold inside the cell, which would prevent the replication, which surprisingly also presented these cells with a new superpower. Even though they were no longer able to reproduce, they could now resist various stressors that would otherwise kill E. coli. For example, they showed stability in hyperoxidative environments, especially in hydrogen peroxide, which normally kills bacteria right away. And that's because this polymer inside of the cells seemed to basically hold the cells together. They were also resistant to some types of antibiotics, the ones that usually target cell walls, and even remain stable in extremely acidic conditions, where most E. coli could not survive. But moreover, they were also then modified to actually contain certain very specific functions. For example, here, by using certain molecular sensors, these cyborg cells could now become responsive to certain chemical inducers and basically become fluorescent in certain conditions. In other words, they could now be used as biomedical markers, but were also completely safe because they could not reproduce. But one of the most remarkable achievements and potentially one of the most useful superpowers these cells seem to contain is their ability to invade certain cancerous cells. In other words, these unusual cyborgs had therapeutic potential. They actually had a chance of maybe creating a new class of medicines, allowing direct and specific treatments that would be otherwise impossible. And though at the moment this is just a proof of concept and they have not been used for anything yet, by itself this is a pretty exciting discovery that we'll definitely follow up on in some of the future videos. But these hybrid cells were just the first discovery. Let's move on to something just a little bit different, but something that's technically even more impressive. A different area of synthetic life that involves re-engineering genetic code itself. Something that's actually never been done before, and in this case something that created a cell with a very unique function. Although first, let's briefly discuss genetics relevant to this particular study. Now, all life on Earth that relies on DNA is based on the same fundamental recipe written in a language that contains 64 different codons. You can see some of these codons listed right here, represented by a triplet of nucleotides. And each of these codons provides instructions for each of the cells in order to translate molecules into amino acids, which eventually, after folding, form proteins, which then build the cells, which then build us. And this system, even though it's universal, does contain certain redundancies. In other words, it's not really perfect. Specifically, many codons seem to do the same thing. Here's a typical chart showing us codons and which amino acids they end up producing. And so here, certain instructions basically add to the same amino acid. And so here, even though this is not perfect, it seems to have worked just fine for billions of years. But for biochemists and microbiologists, there's always been this ultimate achievement. They've always dreamed of finding a way to potentially modify this and turn this even better, or basically perfect this by removing some of the codons. And so there was this unspoken goal. A goal in top-down synthetic biology of creating a minimal living cell by essentially removing non-essential genes, removing the redundancy, and only leaving what's needed. And while quite a few previous experiments were able to achieve synthetically modified genomes by creating something that would still create a cell, and by essentially removing all of the junk DNA. This is of course something we discussed in some of the previous videos. But in this new study you can find in the description, scientists were able to achieve something entirely different. A new synthetic genome, once again using E. coli cells as the basis, with a much more streamlined genetic code containing only 55 out of 64 available codons. And they named this SYN57. And while here the synthetic life could indeed survive, could thrive, and actually created regular cells without using all 64 codons and by only relying on 55. And they did this by reverse engineering E. coli DNA completely from scratch. For example here, 4 of the 6 codons for serine, 2 of the 4 for alanine, and 1 stop codon were removed completely. Or to be more exact, they were substituted with synonymous codons that carried identical instructions. And though this might sound kind of simple because basically they just reshuffled some of the DNA, in reality this took approximately 101,000 individual changes inside the genome of E. coli. And this involved meticulously planning individual changes computationally, and then testing small fragments by using living bacteria one by one, with this incremental approach being crucial in order not to introduce any harmful changes and in order to prevent the cells from dying. And while the implications of this compressed genetic code are actually quite exciting. For example, one major benefit here is that this SYN57 now has more free space to introduce unusual or artificial amino acids, which opens up a lot of opportunities for genetic code analysis, enabling researchers to develop synthetic polymers with properties not found in nature. But that's of course just hypothetical for now. Another important implication in this case is in regards to safety and industrial applications. Since the synthetic bacterium has this somewhat bizarre genetic code, it might be completely illegible to a lot of viruses and can thus resist viral infection. And that's because viruses normally hijack the cell's protein production machinery, eventually destroying the cell in the process. But in this case, if the code is unreadable, and if it contains codons the virus is not used to, a typical virus might not be able to do anything inside. And in this case, this particular resistance could be very important for a lot of medical industries. This might reduce costs and setbacks associated with various viral outbreaks where bacteria like E. coli are used for industrial production. For example, today in pharmaceutical industries, E. coli are used in a lot of production facilities in order to create a lot of important medicine. One obvious example is the production of human insulin, or even production of enzymes like renin used to produce cheese. And so quite a lot of medical facilities depend on E. coli, and quite a lot of facilities can be affected by various viral infections. And so here, by creating something resistant to viruses could dramatically improve industrial production. And at the same time, because this organism is so unique and because its genome is so different, we essentially don't have to worry about biocontamination or this bacterium escaping. Because of its illegible genome, it effectively sterilizes genetically modified bacteria in the context of natural ecosystems. And so even if this bacterium escapes into the environment, it's unlikely to affect natural ecosystems at all. And so here this work exemplifies an incredible genetic achievement that could potentially change biomedical and even food industries for the better. But once again, this is just a proof of concept for now, and none of this has been used yet. And last but not least, we also have to discuss another study, but in this case involving a much more complex cell, eukaryotic cell. Reported in a study you see right here, based on the redesign of a chromosome from a very famous yeast, Saccharomyces cerevisiae. And here this is important because normally bacterial genomes are much simpler, but for eukaryotic cells like the ones inside of us and inside this yeast, the genome is just much larger, much more complex in its organization, and involves multiple chromosomes, making it much more difficult to synthesize or redesign. Now here this is actually based on what's known as CS2 Global Consortium, a consortium whose goal is to design and construct completely synthetic genome based on the genome of this particular species. And this started back in 2006 in order to create a brand new set of instructions for yeast designed by humans instead of evolution. Now normally this yeast has the DNA organized into 16 long pieces, each carrying around 6,000 genes. And in order to make this a little bit simpler, this consortium split up the job, with each of the project groups eventually making one of the chromosomes. And the goal was to basically at some point put everything together, creating this from scratch. And this of course was really important in helping us explore how more complex genomes can be physically changed and improved in order to make a complex organism much tougher or even more useful. And since prior research was mostly focused on bacteria, this was the first time ever a complex cell like yeast was used, with all of the lessons learned in this case eventually being used for plants, animals, or of course humans. And so this team from Australia mostly focused on the 16th chromosome known as SYN16. And for the first time ever, they were able to completely redesign it and removing certain defective parts, basically creating a perfect 16th chromosome. And so despite initial challenges and obviously complexities of this genome, eventually the team was able to create this chromosome and even modify it to properly use glycerol as a potential energy source at high temperatures, which once again when it comes to industries could help us use yeast, for example in the production of various biofuels and pharmaceuticals, or to produce certain industrial chemicals. And here the ability to engineer a eukaryotic chromosome is almost like a quantum leap when it comes to biological sciences. And so by being able to assemble the first synthetic genome from a more complex eukaryotic cell, this sets up a landmark for synthetic biology and the future design of life for very specific scientific, medical, and industrial uses. And because this is a collaboration and a global scientific endeavor, at some point it will hopefully benefit everyone. With the recently held SynCell Global Summit that was in China, eventually bringing together a lot of researchers from across the planet with the aim of establishing a consensus for the future direction of the cell research when it comes to synthetic biology. And so even though there's been a lot of talk about things like, for example, artificial intelligence that's dominating the news, in my opinion, stuff like this is way more interesting and potentially more influential, because this will teach us fundamental understanding of cell biology. It will also help us probe theories of origins of life, potentially create a lot of functional cells and improving biomanufacturing, and provide a true test of understanding of life itself. But obviously this is just the beginning and there are still a lot of challenges. For example, building a fully functional cell is still way beyond our means. Not to mention there are also ethical, biosafety and societal concerns, all of which have to be discussed before we can make this into a fully functioning industry. For example, there are still potential risks that some of these synthetic cells could maybe disrupt ecosystems or could be somehow misused and become harmful, and so here engaging the public and implementing safeguards is also of course something that needs to be discussed. But in essence, in just the last decade or so, there's been this really important paradigm shift when it comes to cellular biology, with the focus being rewriting the very code of life itself and even recreating certain types of code completely from scratch, with a new approach also being bridging the gap between living and non-living, essentially creating a kind of a cyborg. And though obviously the journey in this case has just started, with many fundamental questions still unanswered, based on global summits like this one, and based on a lot of studies that came out in the last few years, all of this kind of hints on this potentially being the future of microbiology. The ability to engineer life from scratch, and the ability to modify pretty much everything in order to make things a little bit more efficient. But exactly what comes out of all of this, and what's discovered next, we're only going to learn in some of the future studies. And so until then, check out previous videos in the description. Thank you for watching, subscribe, come back tomorrow to learn something else, support the channel on Patreon where you can find additional videos without any ads, and can DM me directly, or by joining a channel membership that grants you early access. Alternatively, you can also buy the Wonderful Person t-shirt in the description. Stay wonderful, I'll see you tomorrow, and as always, bye bye.